All right. Well, thank you everyone for coming today. It's my pleasure to introduce Fernando Hartwig, who is a researcher from the University of Pelotas. He has a long-standing collaboration with us here in Bristol. He's done a series of really interesting and important and widely used papers investigating methods, particularly for pleiotrope, robust Mendelian randomization, and a number of other really important methodological developments. So I'll hand over to Fernando. Looking forward to what you've got to say today.
Thanks. Thank everyone for joining today. Thanks for the opportunity to present here. Since you have, a small number of people actually in the room physically, I feel comfortable with taking my mask off so people can understand me a little bit better. So what I wanted to talk about today is to really provide some examples to illustrate this idea that we can use genetic data which is getting increasingly available in epidemiological studies, not only to do like more basic science research, which is typically what more conventional genetic epidemiology studies do, but also to address sort of more directly questions of public health relevance, and you understand what I mean by that throughout the presentation. So this is just a summary of what I'm going to talk about today. So to start things off, let's briefly talk about genetic epidemiology and genetic data. And as always, it's good to start with a few definitions of what we're talking about. So we can understand epidemiology as the study of the distribution of health, disease states, and their determinants in populations. It doesn't have to be human populations, but here we're interested in human populations. This definition of epidemiology was actually elaborated by Cesar Victoria, which is a researcher in Pelotas and was my PhD supervisor. And if we just stick the word genetic in there, then you have genetic epidemiology, which we can understand in a simple way as the study of the frequency of genetic variants in populations or subgroups of a given population and how those genetic variants relate to health and disease. And although it is not strictly necessary to actually collect genetic data to study genetic epidemiology, for example, you can study segregation of traits in families, and this will give you some idea of heritability of the traits. Most modern genetic epidemiology studies do use genetic data and often massive data sets of genetic data, and this is the type of study we're going to be focusing on today. So by genetic data, I mean any direct measure of the genetic makeup of an individual. And by direct, I mean that genetic material was actually collected and assayed in some way. And you can have many different types of genetic variants, and we often talk about single nucleotide polymorphisms, which are very simple. So I think that's why we're interested in them, but it can be of many forms. And in the sort of current state of genetic epidemiology, most studies deal with or have genome-wide genotyping data, which is basically large-scale data in terms of number of SNPs scattered throughout the genome. And as I said, this is becoming increasingly available in several cohorts worldwide because the costs are going down, the technology is developing, and all that. So now I just wanted to briefly talk about what I call standard, although this word is getting a little bit outdated now, but standard applications of genetic epidemiology. And if we look at what most genetic epidemiology studies would do, say 20 years ago or 15 years ago, would be to test the association between one or more genetic variants with a health outcome of interest. It doesn't have to be a health outcome, but when you're talking about epidemiology, we are often talking about health and disease states. And we have like two main sort of classifications of studies that do this: candidate gene studies and GWAS studies, and they differ regarding number of variants, the type of data that you need to do them, the cost and complexity, and the conceptual paradigm that you perform each one of them. In terms of genome-wide association studies, the typical goals of those studies are to identify genetic variants associated with a given phenotype. And this is completely hypothesis-free. So, when you do that, you're really just identifying statistical associations. And from those, you try to gain some new biological insights about, this disease or the phenotype that you're studying. And those insights can lead to practical applications in the future. But those practical applications wouldn't be the sort of primary goal of those studies. The primary goal would be to provide sort of biological insights. And in this sense, they would be more like a basic science kind of study. And these are some characteristics of GWAS studies, millions of hypothesis tests. We have millions of variants, so test the association of each one of them with the phenotype. So you need to do a multiple testing correction, replication due to linear scores to establish associations. And as a result, you need massive sample sizes, which means that you're pretty much required to do consortium-based efforts. Perhaps, before this massive sort of biobanks like the UK biobank, your only option was really to do consortium-based efforts. So talking a little bit about practical impact of genetic epidemiology, as we just discussed, those studies are primarily of a basic science nature. It doesn't mean that they're not useful, it just means that they are not primarily interested in providing more sort of applied results. And what I want to discuss today during the rest of the presentation is ways that we can go beyond this sort of conventional approach to genetic epidemiology and explore genetic data in a way that we can understand sort of health disease processes in a more applied way. And indeed, people here are experts in doing that through median randomization, but there is more to it than MR, as I hope to illustrate with some examples today. So now what I want to do is I will just provide, I think, 5 examples of applications that we can do in this context. One of them is MR, and the other 4 we're gonna see. So the first one is in social epidemiology, and perhaps social epidemiology would be the last field that you would expect me to discuss today when it comes to using genetic data apart from data randomization, of course. And I wanted to talk about ethnic-related social inequalities. Those exist in many countries, including my country, Brazil. And due to many historical processes, slavery, racism, and many other things. And these result as in negative health outcomes being way more common in ethnic minorities. And this is very clear in say, the Pelotas cohorts, for example. So if you want to study ethnic-related inequalities, we need some measure of ancestry or ethnicity. And the sort of conventional way of measuring this in epidemiological studies is to either ask the interviewer how he or she self-identify in terms of skin color or race or ethnicity, or me, the interviewer, I look at the subject and I classify the subject into something. Those measures are both subjective and they're likely prone to systematic measurement error. We actually did some analysis in the Pelotas cohorts where the population is highly mixed. And we see that your socioeconomic position influences how the interviewer classifies you in terms of ethnicity or skin color. So an alternative to subjective measures is to use genetics to understand the ancestry of the individual. And the advantage of this is that first is an objective measure, second is a quantitative measure, So I don't classify two different people, say, as black or white. I have the sort of proportion of each ancestry for every given individual. And this is getting more and more affordable over time. Many people are interested in this kind of service. So when we derive ancestry measures based on genetic data, people often call this genomic ancestry. And we can calculate that or estimate that through a series of steps, and here are the simplified steps. So we need to have a set of genetic variants, often a couple hundreds. If you have G1's data, just like use the whole thing. But you need at least a couple of hundreds of genetic variants whose frequencies differ substantially between populations of homogeneous ancestry, and those are your reference population. So you say you have a set of, 1,000 individuals that are known to have European ancestry, then that's your sort of European reference population. And you can do that for different ancestries. Of course, there are not many, truly homogeneous populations nowadays, but I would say there are populations that are at least less mixed than others, so we do what we can. So what we essentially do is you compare the genetic profile of your proband with the genetic profile of those reference populations, and you use some similarity metrics to estimate the proportion of each ancestry that your proband has. So here are just some results of genetic ancestry or genomic ancestry in the Pelotas cohorts. So here, let's just look at the top row of this figure. Can you see my arrow? Okay. So here we see each one of those plots show the proportion of each one of those ancestry groups. So for example, the first plot here shows the proportion of African ancestry according to self-reported skin color. So we see, as expected, that the sort of median is a proportion of African ancestry is way smaller in people that self-identify as white compared to people that self-identify as black. But we see, for example, that a lot of people with not so low values of African ancestry still self-identify as white. And this relates to the fact that this measure is subjective and, depends on how you understand yourself and things like that. So in this other figure here, we see in this black solid line here, the probability of you self-identifying as being black according to the amount of African ancestry you have. So here in Pelotas, you have about 50% probability of self-identifying as black if you have about 30% African ancestry. So it's quite a low threshold. And if you go, for example, to Salvador in Bahia, which is a completely different region of the country, you have 50% probability of self-identifying as black if you have about 50% African ancestry, which is more like you would expect in a way. So this shows how different self-reported measures mean depending on the cohort that you're studying, even within the same country. So we actually applied this data to several analyses now. This is just one example. We're looking at the association between African ancestry and major depression in adulthood. And here we stratified the analysis according to socioeconomic position in the first, second, and third tertiles. And we see that in the third tertile, we see evidence of higher risk of depression among those who have higher levels of African ancestry. And the more you have, the higher the risk that you have major ancestry in adulthood. And then we ask the question, is this association, assuming it is causal, of course, mediated by skin color discrimination, which is something that we measured in the cohorts, and we saw that most of the association appeared to be mediated through skin color discrimination. So here we're using genetic data to address a question that is essentially social. So going to the second example now in nutritional epidemiology, there are several sources of evidence that suggest that breastfeeding improves cognitive development. And of course, this is debatable, but let's just assume that, this relation is indeed causal, just for the purposes of the presentation. Then we can wonder, why is that? Like, what would be the possible mechanisms for this association? And people often postulate nutritional benefits or psychological benefits like mother-child interaction and, many others. Focusing on nutritional benefits, one of the plausible mechanisms for this effect would be that breast milk is a source of a I wrote long-term, it should be long-chain polyunsaturated fatty acids, which are, important for brain functioning and many sort of neural processes. Here is a summary of the pathways implicated in the endogenous synthesis of those fatty acids. I don't want to go through this. I just want to point out here with the red boxes that at several stages of this pathway, we have this FAD2 gene, you can see here. And I'm just highlighting this because we're going to be looking at genetic variants in this gene, just justifying why we're picking that gene. And the question we want to ask here or to address here is a question that people sometimes formulate as a nutritional adequacy hypothesis. And the hypothesis goes like this. In this first graph here, we have the levels of the fatty acids that you have through your diet or through endogenous synthesis or whatever source, supplements, whatever source you have as a child. And on the y-axis, you have levels of cognitive development. This is a very sort of non-quantitative kind of graph. I'm just illustrating ideas here. And the idea here is that if you increase your level of these fatty acids, you improve cognitive development on average, but this has a limit. At some point, further increasing the levels of fatty acids does not further increase cognitive development. Some people call this law of diminishing returns. And on this relates to the other graph here, where we have sort of 4 subgroups of the population. Those that have a generic profile that favors the endogenous synthesis of those fatty acids. Those are shown here. And the other group is the group that has a genetic profile that is correlated with lower levels of those fatty acids on the other side of the figure. In black, individuals who were breastfed as a child, and in gray, individuals that were not breastfed. And the hypothesis is that if you were breastfed, you can achieve the threshold, the sort of minimum threshold of long-chain fatty acids required for optimal cognitive development. As you can see here, both of the black bars reach the threshold level. However, if you were not breastfed as a child, then you can only achieve this threshold if you have a genetic profile that is associated with higher endogenous synthesis of those fatty acids. So that's a hypothesis. I'm not saying that that's how the world works. That's a hypothesis. So we can understand this as following. Breastfeeding sort of buffers the downside of having a genetic profile related to lower synthesis of those fatty acids. That would be the hypothesis. So we would only see an association between the genetic profile and cognitive development among those who were not breastfed. Because if you were breastfed, then you would achieve the threshold anyway. That's the hypothesis. And people actually looked at this hypothesis in the past. So here is a study that was conducted in ALSPAC using ALSPAC data. IQ was measured, I think, at age 10. And they used a genetic variant in the FAD2 gene that we talked about earlier. And here, GG would be the lowering genotype. So the genotype correlated with lower endogenous synthesis of the fatty acids. So the result that we see here in terms of the association between the genotype, breastfeeding, and cognitive development is quite what we would expect based on a nutritional adequacy hypothesis. For breastfed individuals, the dark gray sort of bars here, IQ seems to be more or less, the same in terms of average across groups. And if we looked at individuals who are not breastfed, then they always have lower average IQ, which is what you would expect. But this is especially true in the lowering genotype, which is exactly what you would expect based on nutritional adequacy hypothesis. However, an earlier study did not see that at all. And in fact, they saw that in the GG group, where we would expect the largest effect of breastfeeding was actually the subgroup where you do not see an association. So we have those two conflicting results. One supports the nutritional adequacy hypothesis and the other one doesn't. So to sort of solve this, we set out to do a meta-analysis. Actually not of published studies, it was like a de novo meta-analysis. So people did analysis from scratch, and we included unpublished studies as well. And here is just the pooled breastfeeding gene interaction coefficient. And we failed to see strong statistical evidence supporting an interaction. Just for curiosity, we did a lot of meta-regression analysis trying to understand this result. And one interesting result that we saw here was that in studies with a larger median duration of breastfeeding, we saw a breastfeeding gene interaction coefficient much larger than in studies with a lower average duration of breastfeeding. But I mean, this is not a hypothesis that we set out to test. This is actually a hypothesis that was generated as sort of exploratory data analysis. The point here is not to convince you that the nutritional adequacy hypothesis is true. The point is just to illustrate that we can exploit genetic data to address a question that is related to nutritional epidemiology. So moving to the third example here, again, with breastfeeding, but now with regards to obesity. Observational studies suggest that, if you were breastfed as a child, then you have a lower risk of being obese in adulthood. I'm not saying this is necessarily true. Let's just go with that for the presentation. So assuming that this is true, that there is an effect of breastfeeding on obesity risk as an adult, a possible mechanism that people have been postulating would be that if you were breastfed, then you will better develop your satiety responsiveness, which of course relates to obesity. But satiety has genetic determinants as well, and one such determinant is the FTO gene. And indeed, many studies suggest that genetic variants in the FTO gene are strongly associated with obesity, and this happens through satiety responsiveness. So we could ask ourselves if there is a possible interplay between breastfeeding and FTO, if we could use genetic variants in FTO to try to understand if satiety responsiveness is implicated in this association between breastfeeding and obesity. So we did that in Pelotas. Here I'm just showing results for BMI, but we did for many other obesity measures. I'm just showing one of them for simplicity. It is interesting that when looking at the association between breastfeeding and obesity, we did not see any convincing evidence of an association whatsoever, as you can see here in this table here that I'm pointing right now. But if we look at the association between the FTO variant that we studied and BMI, stratified according to breastfeeding duration, that we see a much more interesting result. For individuals that were breastfed for less than 1 month, and we use less than 1 month instead of 0 because the prevalence of actual 0 breastfeeding was very small. So for individuals that were breastfed for less than 1 month, we see, quite clear association between the genotype and BMI. But when you look at individuals that were breastfed for at least 1 month, we do see an association, but it's way weaker, way weaker. So this does seem to suggest that breastfeeding sort of buffers partially buffers the obesity increasing effect of this genotype, which would be in line with the hypothesis that breastfeeding influences obesity through satiety. So again, we are studying a nutrition epidemiology question using genetic data. So now, causal inference, but not linear randomization. More specifically, genetic confounding. Genetic confounding is, type of confounding, like any other type of confounding. But what is special about it is that before genetic data was readily available, it was something that was very difficult to deal with. We have a few examples, a few exceptions of this, like twin studies. But for most sort of large population-based epidemiological studies, dealing with genetic confounding was very difficult. But now we have genetic data, we can use it to, assess this possibility in some cases. And to illustrate this, we're going to go back to the association between breastfeeding and IQ. One explanation that people have postulated for this would be that if you have an infant that is more apt, like overall, that infant would find easier to breastfeed and would also have a sort of natural tendency to become smart. So this will be a type of genetic confounding. And we can actually empirically assess this, and that's what we did in Pelotas, using based on results from the SSGAC consortium, the second phase of this consortium, which studied the genetics of stooling and IQ through GWAS. So from their results, we calculated a allele score that's convincingly correlated with the participants' IQ in the Pelotas cohort. And the idea is that this allele score captures in some way this latent construct of aptitude, right? So if breastfeeding is associated with this allele score, then would be concerned that we could have genetic confounding in this situation, right? So that's the results. So in the first table here that I'm pointing at right now, we see the association between breastfeeding duration and IQ in adulthood in Pelotas, and a clear sort of I don't want to say dose response, but as you increase breastfeeding duration, you increase average IQ. And here is the association between the allele score and IQ, and the allele score is standardized to have mean 0 and variance 1. So we can clearly see that the association between breastfeeding and the allele score does not match with the association that we see between breastfeeding and IQ. So this result would argue against the hypothesis that genetic confounding is driving the association between breastfeeding and IQ. We're not saying that this proves that the association is causal, we're just finding empirical results that indicate that this particular alternative explanation is not very likely. Yes.
Is there a power issue there? Is there a power issue there? Because all you're showing on the right-hand side is the association between the allele score and IQ, whereas on the left-hand side you've got breastfeeding and IQ, and clearly the allele score is only going to explain the type.
Oh, sorry, it should be, sorry, I just, it was a typo there. It should be breastfeeding and allele score.
Okay.
No, the allele score is associated with IQ. Here, sorry, I just wrote it wrong. It should be, instead of IQ, here should be breastfeeding.
Okay.
Because this is what we're showing. Breastfeeding duration, and those values here in the blue table, the values are the values of the allele score.
Yeah. I hadn't even twigged that the parts are wrong. That's what I was presuming you were doing. But even when you're doing that, is there not a, how do you know that you've got sufficient power on the right-hand side? I guess the confidence intervals.
Yeah, I mean, first it's important to say that the p-value is, does reject the null, but look at the pattern. That's my point. I'm not, very concerned about the p-value here. I'm concerned about the pattern. Here we see, increasing, clearly increasing. There we don't see that at all. So I would expect at least the pattern to be, more or less the same, at least in terms of direction, for genetic confounding to be a plausible explanation. So that's sort of the rationale for this. So the last example, but not, the least important one, is median randomization. I think everyone here is familiar with it, so I'm not gonna, bore you guys even more with this, but just to mention that in MR, genetic variants are tools, not the object of interest, and we are interested in the effects of modifiable exposure, risk factors, treatments, whatever. So again, this ties in with the topic of the presentation. We need assumptions for that, as you are all aware. And the reason why we care is that if those assumptions hold, then your genetic variant is only going to be associated with your outcome if your exposure causes your outcome. So, that's why we care about that. And, as any instrumental variable analysis, you need assumptions. If those assumptions are violated, then, your results are invalid. In MR, this can happen in many ways. People are now very well aware of it. And, there's actually a lot of people trying to develop methods to make the assumptions more plausible, including myself. So what I tried to do here was not to say that the more conventional approach to genetic epidemiology, conventional genetic association studies, I'm not trying to say that those are not important. I was just trying to present a different perspective on genetic epidemiology, where we can exploit genetic data in more quote-unquote applied ways. And I hope to have illustrated this with some of the examples that I shared with you today. So yeah, that's what I had for today's presentation. Thank you guys for joining here in person, and for everyone who is joining remotely, and I'm happy to take any questions.